So to continue our discussion about DNA, we're going to talk about section 12.2, which deals with chromosomes and the process of DNA replication. Remember back from the beginning of the year when we talked about the different types of cells, that there was two different types of cells, prokaryotes and eukaryotes. Okay, prokaryotes and eukaryotes, their DNA and chromosomes are organized a little bit differently from each other. So first we'll talk about prokaryotes. Prokaryotes, remember, they do not have a nucleus, so their DNA is located in the cytoplasm of the cell, okay, because there's no nucleus. And most prokaryotes, they only have a single DNA molecule, and it's usually circular. So we remember that E. coli is a bacteria cell, and they are prokaryotes. Okay, so this is an E. coli cell here, and you can see inside there is its DNA. It has a single piece. There's no nucleus, so it's just located there out in the cytoplasm. Kind of, if we were to look at that little portion of the DNA, this is what we would see. Now, eukaryotes, they have a nucleus, so their DNA is going to be located inside the nucleus, and it's going to be located inside chromosomes. Okay, and remember we talked about this before when we started our genetics discussion, is that different organisms have different numbers of chromosomes. So for example, humans have 46 total chromosomes, 23 pairs. We talked about fruit flies. Scientists like to work with fruit flies. They have 8 chromosomes. Now, chromosomes, eukaryotic chromosomes, during the cell cycle, especially during interphase, they will be very tightly packed together in a form that we know that we call chromatin, and they will be made up of proteins called histones. Okay, and so you can see there's some histone proteins around here, and this is the DNA strand, and it's wrapping around those proteins called histones. And they form, when they do this, they form something that's called a nucleosome. Okay, so you can see here's our DNA, and it starts to wrap around those histone proteins. It forms a complex that's called a nucleosome. You see the little nucleosomes here. Eventually it will keep coiling, 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 and it will form a chromosome. Okay, so remember that during interphase, during the cell cycle, and interphase in particular, the S phase, the DNA is going to be copied so that by the end of mitosis or meiosis, we will have a full set of chromosomes. And so now we're going to talk about that process of DNA replication. Eukaryotic cells have very long strands of DNA, and so when they're going to replicate, and replication just means that we're going to make a copy. Because the strand is very long, this can happen at several different places along the DNA strand. And the first thing that's going to happen in replication is, in order to make a copy, we need to unwind the strand. And these places where the strand unwinds are called replication forks. And so you see here is where we would say our replication fork is. So the two strands have separated, and now they're going to start to be copied. Like I said before, duplication of the DNA, the process is called DNA replication. It just means that we're going to make a copy. So here's our key concept here. That during DNA replication, the DNA molecule is going to separate into two strands, and then it's going to produce 2 new complementary strands. And these strands are going to follow the rules of base pairing. Remember, A pairs with T, C pairs with G when they make the new strand, and then eventually each strand of the double helix will serve as a template for the new strand. So here's our DNA strand, and you can see that it is unwound. Here is our replication fork, and then each of these strands are going to start replicating. So you can see the original strand is the blue one. It's going to serve as our template. And then there's also a template strand up here. So both of these are going to be copied. And there's going to be an enzyme that's involved in this copying that is here. That is called DNA polymerase. Okay, so again, the first thing that happens is we have to unwind and unzip that DNA molecule. There's step 1. And this job is carried out by specific enzymes. And remember that we talked about the base pairs, the A and the T, the C and the G, are connected by hydrogen bonds. Those hydrogen bonds have to be broken. So then the process of replication. The main enzyme involved in replication is called DNA polymerase. We know it's an enzyme because it ends in ASE, and its job is to make DNA, okay? As remember, polymerize means we're going to make a large molecule, and in this case, we're making DNA. Okay, and the job of the DNA is to, or the job of the DNA polymerase is to move along that strand and add those bases. And as it goes, it's going to proofread so that hopefully there's no mistakes in the new strand.